about bioinformatics, which is complementing the other results. So please.
Hi everyone. I'm a bioinformatician, but I have a background in physics. I worked in wet lab before, but I'm doing only computer stuff right now. And today I will talk about pathogens, the ones that we approach in the lab. Actually, it's more about sequencing data, and we look at the microbiomes, and for those we especially look in viruses, bacteria, and fungi, but not archaea. But we also can look at archaea if we deal with 6-ms or shotgun genomics data. We usually do whole genome sequencing, but we also do 6-ms amplicon sequencing and shotgun genomics in the lab. This is the infrastructure that we have. We have two sequencing systems, MySeq and NextSeq, which we use for short read sequencing, but we also do long read sequencing with MinI. And for the approach, starting with the raw data, we do reference-based assembly and de novo assembly of the data. So we will have consensus sequences of the genomes that we look at. And I will start with the viruses. We have some matrices that we use because of the fact that the viruses are highly mutated. We have some substitution matrices that we use for the analysis of the alignments. And why do we use that? Is because we have different genomes. In this case, we have some HCV genomes in different fibroses, and we have multiple alignments, and to look into the mutational profile to see how those mutations affect the virus fitness, we can do some variability profiling based on the mutations that we have. And we have different regions that are highly mutated or different regions that are conserved, like these ones that don't have any peaks. And this is important to look at because of the fibrosis cases for the patients that we have the samples isolated from. These mutations are also important to see how the infection occurred and what are the events for the infection. And based on the multiple alignments of the sequences, we can do phylogenetic appearance to see how these viruses evolve. And in this case, we have some data for nasal cases, But also based on the mutational profiles and the variability, we can use that information to map it on the 3D structure of the proteins. And here you have a 3D structure for the HIV integrase. With blue, you have the most conserved regions in the integrase, and with red, you have the variable parts. And this is important because we can, based on that, we can see where the mutations occur on the 3D structure, and we can tell afterwards if a particular mutation has any propensity for affecting the binding of a drug. Like in this case, artegravid it binds in this specific site where you have a magnesium ion. So you can see that here is it's really conserved, but we also have some mutations in the nearby region. So this is important, and it is important to look at the binding sites and design new drugs for the infections. We have some papers for that. So I will get next to bacteria. So we look at antimicrobial resistance, and we had some previous research grants for surveying different parts in the country to see how the antimicrobial resistance is spread. And we also have a new project right now, Center of Excellence, that will start this year, and my colleague Hilda will talk about more on Saturday. And to resume, antimicrobial resistance is a global challenge, and estimates are not so good. Actually, the estimates tell us that in 2050, the mortality rates that come from pathogenic bacterial infections will overcome the numbers of cancer. And if you look at Europe, for instance, we have in the Eastern European countries high prevalence of pathogenic pneumonia, Entomobacter species. And in Romania, we can see that in the last few years, the prevalence increased with more than 200% on Klebsiella, for instance. But for other pathogens like Pseudomonas and so on, we have the same problem. And with our surveillance systems we want to address, we need to approach One Health systems. But for the moment, we focused only on human health and environment. We intend to collaborate with people from veterinary institutes, and therefore we will have a more detailed One Health approach. The data that we have is isolated from hospitals, from wastewater treatment plants, from the influent and the effluent from the wastewater treatment plants, and also from the surface waters that are in the proximity of these treatment plants. And these are the cities in Romania that we surveyed so far, but we also intend to cover more ground on country. And we are focused mostly on escape bacteria and different ways for bacteria to fight against antibiotics. And these are skinned in this image. Different approaches. We look at multi-locus subtypes. We look at specific housekeeping genes for each species, and some MLSTs are more pathogenic than others. We look at mobile genetic elements. We look at instruction sequences, transposomes, phages, plasmids, and also to look at genomic islands. There are multiple types of genomic islands, but we are mostly interested in pathogenicity islands, which carry variant vectors, and also on resistance islands that carry a lot of antibiotic resistance genes. We also look, if we have lots of genomes, we look at the pangenome. So we study the genes from all the genomes that we analyze from a specific species. And we are mostly interested in the accessory genes that shape the plasticity of the species. And this is a pipeline that I built in the last few years. So starting with raw reads that we get from the sequencing instrument. We do, as I said, de novo assembly and reference mapping. And based on each approach, we can do different types of analysis. And we can also look at mobile genetic elements like the ones that I discussed. So based on the data that we have so far, you can see here some distribution for the subtypes on Acinetobacter baumannii, on E. coli, and Klebsiella species. And we also have some MLST graphs to see which subtypes are shared between different sampling places and which are unique to a certain place. And we see higher variability for E-coli compared to Acinetobacter in terms of MSC. We'll also look at the most abundant genes. So here you have some of the most abundant genes for each species of we looked at. These are species that we have a lot of genome sequence for them. Aside from antibiotic resistance, we can also look at biocide resistance, and in here you can also have heavy metal resistance. You can have resistance given by use of insecticides, fungicides, and so on. So even if some bacteria didn't interact with an antibiotic, it can acquire antibiotic resistance due to the presence of biocide resistance genes. And we have some other graphs in which we look at the antibiotic resistance genes or the insertion sequences in different cities, in this case. So the ones with pink in the middle are the genes that have been found in all these cities, while the ones in purple are specific and are unique to a specific city or shared between only two cities or few more cities. In terms of fine genomic analysis, we can use the information in each genome, and we can look at the core genes that are found in all the genomes. But as I said, we are mostly interested in accessory genes to see which features are designed by specific accessory genes. And we can also look at the metadata based on the genome that we analyze. It is also important to see how these genomes cluster together. And in this case, we have some approximately 2,000 Staphylococcus aureus genomes. Most of them are from human, but some of them are from animals, and we want to see if some specific features are designed for animals in terms of antibiotic resistance, while some others are specific to humans. We also analyzed bacilli series group species, also for phylogenetics. And some other approach, which is really important when looking into more details for the genes in the genome, is the context. So we designed a pipeline that looks at all the genes in the genome, and we tell the pipeline if the genes are in less than 1,000 base pairs to add a tag. And this tag will say that these belong to a cluster. And the cluster breaks if some genes are in more than 1,000 base pairs. So here are some, this is the context for beta-lactamase. It's called OXA72. And we can see that in different genomes from different countries in Europe, we can see different genes that are organized differently in the genome between countries. So this is important if we focus on one specific gene. We can also compare mobile genetic elements or any other gene between used waters from the wastewater treatment plants and the ones from the environment, or use waters and clinical to see if some features are spread in the environment from clinical isolates. And we also can compare by years, and in this case we compared some insertion sequences between 2019 and 2020. And I don't know if it's correlated with the SARS-CoV-2 pandemics, but in 2020 we noticed a decrease in these insertion sequences in bacteria. I don't know if it's due to more care for hygiene, but we notice some differences, and this is not the only one. We can also look at other potential vectors for transmission, like pages. And in this case, we analyze some pages from bacteria, and we can look at the genes. All the genes in each page are shown as arrows, and the ones in red are the core genes that are really important for the page, and the other ones in blue are accessory genes in the page. And we can also look at the cell wall, especially in gram-negative bacteria. We can look at the elements that are qualified by the bacteria for the cell wall, like peptidoglycans, lipid proteins, and so on. And we also found in our genome some resistance islands, like these two cases, which have approximately, this one is 45,000 base pairs, and we found it with an identity of 99%. And we can also infer function based on the pangenomic analysis. And we have some papers that we published regarding this. Regarding the 6-next anticonn sequencing in shotgun, we have done some sequencing on environmental samples, and we looked at the taxons and how they split between different regions, but also based on shotgun, we can obtain all the genomes from that specific community, and then we can do the same analysis as we have done for whole genome sequencing. And in this example, we have analyzed a cohort of healthy people, mostly from a rural area, and we noticed some antibiotic resistance genes in their genomes. With red, with yellow, we have biocide resistant genes, and with blue, you have mobile genetic elements. And we have noticed some correlation between different types of genes, or different genes that respond to different antibiotic categories. And also, in terms of metagenomics, we have done 6-next sequencing on some on the microbiomes from Drosophila, aging Drosophila with parting gene overexpressed, and we notice some changes in the microbiome of plants with aging. And the last part of the presentation is on fungi. Fungi also, fungi in general are really important for human health. There can be good fungi, but also bad fungi. The ones we've read show the lineages of different fungal species that are pathogenic and how they evolve. So such a segment tells us that we have a 0.1 amino acid substitution per each site. So we notice that some fungal species evolve rapidly with a lot of mutations compared with others like Antidabrata in this case. The WHO, not the band, designed the fungi in 3 main risk categories. So we have the critical one, the high and the medium one, and some of them are associated with the environment, while some are human commensals. The most pathogenic ones are Aspergillus fumigatus, Antholoris, Candida and Phytococcinoformans, but these ones, like Candida tropicalis or Nacrocnichus glabratus, are also really important for the human health. The fungi don't have the same evolved mobile genetic element machinery like in bacteria, but they have different traits that are important um, for the evolution and their resistance to antifungals. You can look at variation in ploidy of the fungal genomes. They also have some transposons, like in eukaryotes, like humans have the copy and copy paste and cut and paste retrotransposons. So we find similarities in fungi as well. But we can also look at copy number variation, for instance, and this happens a lot in fungal genomes. Regarding the most relevant Candida species, Candida albicans, Glabata, Cartulosis tropicalis, and Crustei, we notice that the spread has overcome a lot, a big part of the world. But in this case, we've read the spread of Candida auris, which is highly pathogenic. It's split in 6 different clades, and the clade number 1, which is also found in Romania, has the highest mortality rates. It can reach 68% mortality rate in humans with other pathologies. So we were the first ones in Romania to sequence Candida auris, and we have found in the genome that we sequenced, we have found anti-fungal resistance genes for different classes of fungals. And we noticed different mutations that are also related with high pathogenicity. We looked at the variants for each genome and each chromosome, because Candida auris has like 7 chromosomes, and some of these mutations occur in fungal resistant genes. We have done phylogenetic analysis, and we noticed 2 different entrances in Romania for the fungal infection, for the Candida auris infection. One is from the US and one is from Western Europe. And for other fungals like Candida tropicalis, we also look at the antifungal resistant genes, the ones with green, the virulence factors that they have, and other genes that are related to resistance. And here you can see a table with copy number variation. So these fungal species have multiple copies which occur not only on, they're not randomly distributed across the chromosomes, but some resistant genes have multiple copies in different chromosomes. And for Candida tropicalis, which is a diploid species, we also need to look at the copies for each chromosome and see if some the variants are on copies or not. And we also look at the synteny to see how each element in the genome is aligned compared with other genomes. And last but not least, we also look at the 3D structure of different antifungal resistance genes, and also the antifungals that are in 3D structure through X-ray crystallography. And we also develop computational methods. This is an example of a computational method that we developed that does all the or some parts of the things that I discussed, like MLSC distribution, QC quality control of the assemblies, virus spectral prediction, antibiotic resistance predictions, and also pangenome analysis. And some of the challenges that we face are funding, specialized personnel, computing power, because most of the analysis that we do need a lot of computing power, and We have problems with low data and low metadata in the databases, existing databases that we use to compare the genomes. So there are different challenges that we face. And I also want to thank my colleagues from the National Institute for Infectious Diseases and colleagues from the University of Bucharest, and my collaborators from Birmingham, Oxford, from Göteborg, and also from Pretoria. And thank you for your attention.
Please, the questions.
I wanted to ask a bit more about the mobile gene elements. How do you identify them? Is there a separate assay, and where does that fit into the pipeline?
We don't, if you mean wet lab assays, I don't know. I only use databases, sequence databases. So we use the sequences, the reference sequence from different databases, and we compare our data with those sequences. And
yeah, what I meant is for the identification of those mobile gene elements, do you do that also de novo, like you do with what's it called? Assembly, like with a genome assembly? Or do you use references for that?
We use references.
Okay.
But we can also look into de novo, but for that we need to do functional profiling, inference, to see if some of those elements are similar with the known ones from different databases, and for that we can also do proteomics analysis based on the 3D structure of the proteins that they adapt.
Mm-hmm.
Any other question? No? Yes, please.
I like very much your thought. Did you ever check on Epstein-Barr virus in Romanian population?
No. No, we usually work with HIV, HTLV, influenza, measles, SARS-CoV-2, HCV, HBV, but we haven't used Epstein-Barr.
Epstein-Barr virus is linked to chronic lymphocytic leukemia. Usually it's a strain who lost 23 out of 25 microarrays from the genome. This is a bad strain, which is more frequent in the Mediterranean place than in Israel. A lot of CLL in this population, Israel and Mediterranean, in Japan and in China, where the strain is complete with all the genes. They barely found or rarely found chronic lymphocytic leukemia. So I would say it's very interesting if you sequence, because in this part of the Balkans, theoretically, we have most of the variations that can be. So it's interesting to find the
Yeah, we usually, what we sequenced were the samples from patients that were hospitalized in our institute. So this is how we work with different viruses. But I think we didn't have any patient with Epstein-Barr virus. But yeah, I would put in mind.
all people in this room, everybody
Just test anybody, yeah.
Okay.
It's a kissing disease. So if someone doesn't kiss, still can take from so these 99, I would say 100% will say I have it because
Okay.
But specific strains are very
So it's similar with herpes virus.
EBV, it's absolutely ubiquitous. Like Kaposi sarcoma virus. Kaposi sarcoma virus in epidemiology books are 25% of the population. When we check on microarrays from Kaposi sarcoma in plasma of sepsis patients, we found up to 75%. So it's a much more frequent infection who do not induce transcription of the full genome of the Kaposi sarcoma, but of microarrays. Who are doing bad things for the individual because they predispose them, they are risky for septic shock. And to septic shock, 25% of patients die in any institution in the world, including in MDR. 1 out of 4, we are losing them because there's no way to do anything with this. Kaposi sarcoma is also important to sequence if you have an open route to emergency development.
Sure, thanks.
good suggestion. Any other questions, please? If not, thank you once more.